Kumusta ang plant design defense niya? Tapos na ba? Yung mga tapos na?
Further.
Kailan ba kayo ibita? Malapit na kayo, di ba?
Sir.
Okay lang ba ako sa inyo? Hindi ako choppy sa inyo. Choppy ka kasi bukas. Kayo ba namili ng panel niyo? Kayo namili ng panel niyo? Inalaw kayo ni Sir Just na mamili ng panel? Or si nag-assign? Si Tristan na nag-assign? Kayo namili ng panel na Ibita? How do you get your how do you have your panel? Did they assign it to you or kayo namili? So yes. Ilan na? 16. Oh, pwede. So I'm going to share my PowerPoint. So today we're going to discuss DNA replication and then transcription. Transcription is RNA synthesis, tapos protein synthesis. So medyo relevant siya kasi sino sa inyo ang nagpa-vaccine ng Pfizer o kaya Moderna? So yung technology ng Pfizer at saka Moderna ay mRNA. No? So makikita niyo mamaya dito yung mRNA kung paano siya nagpo-produce ng protein sa protein synthesis. Let me share my PowerPoint. Okay. I don't know kung sa usually sa organic chem na-introduce na yung mga nitrogen base, So we have 5 nitrogen base. Hindi ko alam kung ito lang 'yan, pero these are the common nitrogen base, So I don't know if you have heard of adenine, guanine, and then cytosine, no? So you can find this nitrogen bases both on DNA and RNA. Narinig niyo na rin ba yung RNA? Siyempre, narinig niyo na rin yung RNA at DNA. Di ba yung DNA? It has something to do with genetics, with heredity, di ba? I don't know, probably high school, So DNA at saka RNA. DNA is deoxyribonucleic acid, Tapos RNA is ribonucleic acid. Tapos merong nitrogen base that you can only find in DNA. That's your thymine, Tapos meron din namang sa RNA lang meron, uracil. Basically ang nangyari sa RNA, walang thymine, merong uracil. Okay? Tapos yung pinaka-backbone, parang dun kung saan nakakabit yung mga nitrogen bases are sugar and phosphate. So magkaiba yung sugar sa DNA at saka sa RNA. Sa RNA, we have this ribose. So ribose is a 5-carbon sugar. Alam niyo naman ng sugar, meron kayong OH. May OH kayo bawat carbon, Tapos yung sugar sa DNA, tinanggal yung OH sa carbon number 2. That's why you have 2-deoxyribose. Yun yung kaibahan ng sugar nila. Pero both of them, the RNA at saka the DNA, ito parehas yung backbone. Sugar phosphate, sugar phosphate. Na-imagine nyo ba? Sugar phosphate, sugar phosphate. Tapos nakakabit yung nucleic base dito sa sugar. Okay? So ang DNA, double strand siya. Okay? Ang RNA, single strand lang. Okay? So you're familiar with this. Yung iba, single ring lang. So these are your purines. And these are the purines. Yung double ring? Yung double ring purines. Tapos yung single rings are the pyrimidines. I don't know kung naabot nyo sa organic chem. So yun. So para matandaan nyo na may uracil sa RNA, ito yung memes. So baby, I wish I was RNA so I can have you. Basta may mga memes yan, lalabas yan sa board exam. Okay? U is for uracil, Kasi sa RNA lang yun meron. Why am I explaining this? Okay, these are the two types of nucleic bases. Your purines. So purines are adenine and guanine. And the pyrimidines. So the pyrimidines are cytosine, thymine, and uracil. So sa DNA, what you have is your adenine, guanine, cytosine, and thymine. Sa RNA, what you have is your adenine, guanine, and uracil, and cytosine. Tapos you're familiar, tandaan niyo pa yung pairing, Gabi Concepcion, kilala niyo pa ba si Gabi Concepcion? So ibig sabihin ng Gabi Concepcion, di ba, GC. So guanine pairs up with cytosine, sila yung nagpe-pair up, Tapos altantai, kilala niyo pa si altantai. mga mnemonics ko. So adenine pairs up with thymine, makikita niyo yan. Ganun yung pairing, So siyempre sa RNA wala namang thymine, so wala kayong altantai. Instead, adenine pairs up with uracil. So yung pairing up, as you will observe, purine and pyrimidine. H-banding yan, makikita nyo mamaya. So purine at saka pyrimidine yung nagpe-pair. Okay. So we call this nucleoside. When you have the sugar, of course, your ribose or deoxyribose, tapos may nakakabit sa kanyang nitrogen base, ang tawag dyan ay nucleoside. So maraming application ng nucleoside. Generally, ito yung standard ng mga antivirus agent. Siyempre, ang ginagawa lang nila, basically the technique pag antivirus, binabago lamang yung structure, Kasi 'di ba may 5 naturally occurring na nucleic bases. So ang ginagawa, medyo binabago lang nila yung structure nito, Parang structural analog lang siya. Kasi basically, this will be delivered, to the virus. So magiging part siya ng chain nung virus. Kasi virus are proteins, di ba? So pag halimbawa merong isang irregular, non-standard na nucleic base na napasingit sa kanya, eventually mamamatay yung virus. So ganun yung technique ng ng mga antivirus. Yan. We have lots of antivirus. Hindi pa pala yung antivirus. These are your nucleosides. So halimbawa, adenine siya, tapos meron siyang kasamang, what is this? This is your ordinary ribose, may oxygen. So we call that adenosine. Yan. So pag nagtrabaho kayo sa industry, kasi I happen to work on a nutritional company. Di ba, alam niyo naman yung Bona Kid, meron siyang 1, 2, 3, 4, 5, meron siyang 5 nucleotides. Kita niyo naman yung commercial nun, di ba, yung mga batang palaban. Then makikita niyo, raw mat namin yan, adenosine 5-phosphate. So, you might be thinking, sa lang yan, sa libro lang yan, sa school lang yan. Actually, kayo, pag lumabas na kayo sa industry, makikita niyo talaga yung mga pinag-aaralan natin lahat. So, this is adenosine. Ito yung kanyang counterpart na deoxyadenosine. Usually we have this one. Ito yung meron kami. Kasi these are deoxynucleosides. So actually hindi pa rin naman to, magiging adenosine 5-phosphate pa. Pag kinabitan mo kasi ito ng phosphate group, nagiging nucleotide. Ito nucleoside pa lang, So that's why you're not going to read sa label niya, you're not going to read adenine, you're going to read adenosine 5-phosphate. Kasi nucleoside yun, meron na siyang kasama sugar. You're not going to read guanine, you're going to read guanosine. No, sa thymine naman, ito, no? You're not going to get uracil, you will read uridine. Yan, this is an example of antivirus agent. Acyclovir. Parang remdesivir, di ba? Ganun. Sounds like remdesivir. So it is primarily used for the treatment of herpes, no, chickenpox, tsaka shingles. Pag dalawang beses kayo nagka-chickenpox. So other uses includes prevention of cytomegalovirus infection at saka Epstein-Barr virus. So kita nyo to, mukha siyang nucleic base, and then ito yung sugar niya. Ito yung pinalitan. So ganun yung structure ng ng mga antivirus. They are structural analog ng mga nucleoside. So aside from acyclovir, ito yung madami, Ito yung madami pang antivirus. Okay. Tingnan niyo na lang yung PowerPoint. Currently meron kaming may naibigay akong topic sa thesis ngayon, yung third year ngayon. I let them use Autodoc. So Autodoc is a simulation software. Ida-download mo yung structure ng virus. We use SARS-CoV-2, yung nagkakos ng COVID-19. Dinownload namin yung SARS-CoV-2. Tapos we are trying to measure kung gaano ka-effective yung different antiviral drug. Kung mapapatay niya ba yung mapapatay niya ba yung virus. Meron kaming isang ganung topic sa thesis. Sayang hindi ko agad naisip nung panahon niyo Ibita. Panlaban sana Lang hindi rin natuloy. Pero baka kung yun yung sinubmit natin, gusto yun ni Sir Digos kasi biochemical engineering yung head, no? Si Digos. Oo nga, pero malay nyo kung yung sinubmit natin ganun, biochemical, baka tinuloy niya pa rin, Kasi gusto niya yun. Biochemical kasi si Sir Digos, yung head ng Okay, so pag nilagyan niyo ng phosphate group, yung nucleoside, so phosphate plus nucleoside, you're going to have nucleotide, no? So yun, phosphate plus the ribose plus the nitrogen base, you have the you have the nucleotide. Sayang nga, hindi na rin kayo ngayon nakakapag tawag dito OJT. Dati ang dami-dami namin na ipapasok na OJT sa White at saka sa Nestle. Diyan yung makikita yung mga adenosine 5-phosphate. These are nucleotides, So, makikita nyo talaga yan, how do you call this? Sako-sako, no? Tapos nilalagay yan dun sa mga gatas na pinapadede sa bata, So, yan. These are nucleotides. So, 5 yan, adenosine 5. This is adenosine 5-phosphate, Okay. So ito yung primary structure ng DNA. As you can see, nakikita nyo naman sugar, phosphate. Ito yung sinasabi ng sugar phosphate. Sugar phosphate. And then you have the nucleic base. So you have CGAT. Usually we just refer this as CGAT. Kasi yung haba-haba naman ng adenine, thymine. So we usually use CGAT. Yan. Tapos ito yung sinasabi ko sa inyo, Gabi Concepcion. Probably si Gabi Concepcion kilala nyo pa, Parang lumalabas pa yun sa TV. Pero sabi, hindi na nga daw kinanood ng TV. Sabi nung studies, hindi na nanonood ng TV yung mga bata ngayon. So G pairs with C. Yan lang naman yung ibig sabihin, like this one. G pairs with C. Tapos altantai, A pairs with T. Parang adenine pairs with thymine. Ganito yung DNA, So pag-expect nyo, pag merong adenine doon sa kabila, isang strand, sa kabila, ang nakakabit sa kanya ay thymine. Actually, ginagamit din yan sa DNA replication. Ganyan yung itong pairing na to. Kaya when you have a DNA, pag ni-replicate yun, you're going to have the same DNA. Yung pairing na yan, yung Gabi Concepcion Altantai. Lala nyo pa si Altantai. Parang director na siya ata ngayon. Well, you can, of course, you can always have your mnemonics, Pwedeng mga Korean guys and Korean girls. Yan. Bakit sila nagpe-pair? It's because of the H banding na napo-form. No? So, halimbawa, guanine and cytosine, ito yung Gabi conception. You're going to form 3 H banding. Ito yun. Ano? O, syempre, H banding is H with O or H with N. And then another H with O. Ano? So that's why the DNA is a double helix yung kanyang shape. Ano? So, because you have 2 strands, tapos it pairs up the nucleic base, no? The nucleic base pairs up. So that's why you have a double helix shape. Okay? And then sabi ko nga sa inyo, we're going to take up central dogma today. So central dogma refers to DNA replication, no? Production of mRNA or RNA synthesis. This is also known as transcription. Protein synthesis. Protein synthesis is also known as translation. Okay, so itong tatlong process na to, this is known as the central dogma theory. Yan. Di ba pag merong sa Facebook merong evil central dogma. Evil central dogma pag ganun yung direction ng arrow. The protein produces the mRNA, and then the mRNA produces the DNA. Remember, that's the conspiracy theory sa vaccine. Mababago daw yung DNA ng humans pag gumamit ka ng mRNA vaccine. That's the evil central dogma. Okay, so let's take a look first at the DNA replication. So sabi ko naman sa inyo, yung DNA, dalawang strands, no? So these are the 2 strands, ano? So ang unang step sa DNA replication is you have an enzyme, no, or a protein. Ang pangalan niya ay helicase. So yung helicase, basically hinahati niya yung DNA into 2. Kasi paired up yan, di ba? May H banding. So hinahati nung helicase yung DNA into 2. That's the first step. Tapos yun, merong mga protein that binds dun sa mga nucleic base, no, sa mga nucleic acids para hindi sila mag-paired up ulit, ano? So yun, you have single strand binding protein stabilize the unwound parental DNA. Tapos yun, pagkatapos you have DNA polymerase. So the DNA polymerase ang ginagawa niya sa solution, you have nucleotides. So basically binabind niya yung nucleotides dun sa DNA strand. So pahaba ng pahaba yun. Binabind niya dito yun. So the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Actually, specifically, this is DNA polymerase 3. Ito nagpapahaba nitong sa leading strand. So meron kasing leading at saka merong lagging strand. Ibig sabihin pag leading, siya yung unang nabubuo. Tapos this one, ito yung huling nabubuo kaya lagging. So DNA polymerase 3 yan. Dire-diretso lang yan, sinusundan lang niya yung helicase. Tapos, dito naman, ito kasi kaya mabagal siyang mabuo kasi konti-konti lang. So, ito, number 4. The lagging strand is synthesized continuously. So, primase synthesize a short RNA primer. So, primase muna yung nag-a-act dito sa lagging strand tapos bumubuo siya ng RNA primer. So, RNA ito, itong red RNA siya. Hindi siya DNA. Tapos, dudugtungan siya ng DNA polymerase 1. Dudugtungan siya ng RNA polymerase 1. So, ang tawag dito is the Okazaki fragment. Kasi siyempre, a Japanese guy named Okazaki discovered merong ganitong step sa DNA replication. This is the Okazaki fragment. Okay, na-imagine nyo sa laging strand kung ano nangyayari? So, after the DNA primer is replaced by another DNA polymerase. So, DNA polymerase 1. I think DNA polymerase 3 na naman to. I-replace niya tong mga RNA, itong red na to. I-replace siya ng DNA polymerase 3. Okay? Tapos pag marami na diyang Okazaki fragment, it will be acted upon by ligase, DNA ligase. Pag dudugtong-dugtongin niyan to form the second DNA sa laging strand. DNA ligase. So yun, ganun yung kwento ng DNA replication. Do you have any question regarding DNA replication? Yan, memes na naman. I wish I were DNA helicase so I could unzip your genes. yung DNA ano? Genes is your DNA. Are you DNA polymerase 3? Because you're elongating my leading strand. Nanonood ba kayo ng ano? Nanonood pa ba kayo ng engineer to eh, di ba? Engineer si ano. Okay, so ito lang naman ano, para lang matandaan nyo kung pag sa leading sa leading strand, yung DNA polymerase dito is DNA polymerase 3. Yun lang naman yan, ano? Sa laging strand, yung DNA dito is DNA polymerase 1. Pero eventually, pag papalitan na yung RNA fragment, mag-a-act din dito yung DNA polymerase 3. And then finally, you have a ligase. Ano? Question? Primase yung naglalagay ng RNA primer. Okay, so we have here the summary of DNA replication. So, helicase unwound the DNA. Tapos DNA polymerase 3 starts to elongate the leading strand. Imagine nyo. Tapos RNA primase attach an RNA primer to the lagging strand. Nasa lagging strand na tayo. Ano? DNA polymerase 3 starts, should be 1. I think 3 nga siguro. So DNA polymerase 3 starts to elongate the lagging strand called the Okazaki fragment. So 1 yung yung magpapalit sa RNA primer, ano? DNA polymerase 1 replaces the RNA primer with DNA. Ibig sabihin DNA nucleotides, ano? So pag may mga uracil doon papalitan. And then finally, DNA ligase connect to Okazaki fragments. Do you have any question? So galing sa isang DNA, magiging dalawa na sila. That's why this is DNA replication. Okay. So next is transcription. So transcription is RNA synthesis. So we have 3 steps, initiation, elongation, at saka termination. So basically sa initiation, merong so ang RNA galing din siya sa DNA, no? So pag nahati ang DNA, yung isang strand dun magsisimula yung RNA synthesis. So the first step is meron kasing parang code, ang pangalan pag yung pag yung kanyang sequence ay TATA, dun kumakabit, dun kumakabit yung TF means transcription factors at saka yung RNA polymerase. Dun sila lahat kumakabit, dun sa TATA. You means thymine, adenine, thymine, adenine. So pag merong ganung code, doon nagsisimula yung RNA synthesis. So doon umaatake muna si RNA polymerase pati yung mga transcription factor codes. When you say transcription factor codes, these are different proteins, And of course, RNA polymerase is an enzyme. So parang yung mga TF, yung mga transcription factor, these are also mga coenzymes yun, So kakabit muna sila doon. That's the initiation. Magmi-meet muna yung si RNA polymerase tsaka yung mga transcription factors. Tapos elongation, syempre yon, lakad na tayo, no? So paglakad, ang ginagawa si RNA polymerase, kinakabit niya na yung mga nucleotides, no? So kasi nga RNA polymerase siya, pati yung yung uracil, na na uretidine, no? Pati yung uretidine kinakabit niya, no? Pero hindi siya tumatanggap ng thymine. Kinakabit niya dito sa DNA. Okay? So that's the second step, elongation. And then finally, yung termination naman, meron tayong tinatawag na poly-A-tail. So parang adenosine. So yun naman yung signal na tapos na ang RNA synthesis. So pag nakita yun ng RNA polymerase, hihiwalay na siya dun sa sa DNA, no? Tapos you will have your RNA, you can recover the RNA polymerase pati yung TF, tapos meron pa yung sigma factor. Ganun yung RNA synthesis or transcription. The other word for RNA synthesis is transcription. Yan. Ito yung detail, ano? Marami kasing transcription factor. Actually, madali lang naman yan. Transcription factor, this is 2, Roman numeral 2, ano? Transcription factor 2B, 2A, 2B, may E, may F, may H. Tapos this is the RNA polymerase 2. So nandoon sila sa loob ng RNA polymerase 2. Ano? This is the holoenzyme. Tapos mag-a-attach sila dito sa TATA, ano? Sa Tata box, TATA box. I think this is a TATA box. Pag nasa abroad na kayo, huwag niyo kayo magbabasa ng Tata. Filipino 'yon. It's very Filipino. Diba? EDTA. That's very Filipino, ano? Kasi dati yung mga foreigner namin, mga consultant sa Wyatt, they never called EDTA talaga. They never called that EDTA. That's very Filipino word for EDTA, you know? So this is the TATA box. Mas tama yan. Pag nanood kayo yung YouTube, makikita nyo, TATA box yan. Ayun, so lalakad lang, ano, these are your nucleotides, NTPs, ano? So yan, lumalakad na, elongation na yan, ano? So ito yung RNA. So as you can see, pahaba lang siya ng pahaba, no? As the RNA polymerase and the transcription factor goes on with your DNA. So that's it. No? Pag elongated na, so finally makikita nila yung poly-A tail, ano? Ito yung poly-A tail. So pag nakita nung DNA polymerase nyo na mayroong poly-A tail, ibig sabihin A, maghihiwa-hiwalay na sila. No, hihiwalay na yung RNA polymerase, pati yung mga transcription factor, pati yung RNA, no, na nabuo natin will be released. Okay, so this is the third part of the RNA transcription. Okay, so do you have any question regarding transcription? So ito yung first step, TF2D with TBP. This is your transcription factor, attached to TATA box. This is another protein yung TBP, Tapos RNA polymerase with sigma factor. So this is another protein attached to TF2 and the DNA. So ito pala muna, actually ang Tata box naman nasa DNA na rin naman. So basta ito yung initiation. Kakabit sa DNA yung RNA at saka yung transcription factor. That's the short of it, Tapos yun, after that, the transcription initiation complex is formed. You have your RNA polymerase and the transcription factor, Tapos yun, lalakad na siya, the RNA polymerase will elongate the RNA using nucleotides. So, kukuha na siya, nasa solution lang naman yan, yung mga nucleotides, tapos idudugtong niya na dun sa RNA, And then finally, pag nakita niyo yung poly-A tail, you have the AAA codon, the RNA polymerase detached from the DNA. Do you have any question regarding transcription? Wala naman. Okay lang? And then finally, so ito yung pinaka importante, Ito yung man of the hour, process of the hour, So ito yung mayroong mRNA, So, 'di ba? Ang mRNA product din siya kasi it's just another RNA, So product din siya ng transcription, So makikita niyo naman sa Wikipedia kung paano ginagawa ang Pfizer na vaccine, yung anti-COVID vaccine, They use E. coli, They use E. coli to produce the DNA. So it's a modified E. coli. Tapos that's isang planta yon. Tapos you have another plant. Yung Pfizer nakalagay sa Wikipedia. Yung susunod naman na plant is where they produce the mRNA from the DNA, from the E. coli. Ganun. Tapos yung third plant nila, doon nila pinoform yung solution. They use parang fats, like lipids ang term nila ginagamit nila. They mix the mRNA produced from the second plant, parang they dilute it using lipids. And then yun, diretso na yun, for injection na yun. Kaya mabilis ang Pfizer. Kaya lang ang taas pa rin talaga ng demand sa Europe kasi yung mga bata tinuturukan nila. Pero actually relatively lately, medyo marami nang dumarating. Ano? It's relatively new technology, yung mRNA. Ano? Bago lang yun. May expect nga nila it will win the Nobel Prize pag nawala na yung COVID. Okay, let's take a look at translation. This is the protein synthesis. Ano? So let's see it this way. Pag in-injectionan kayo ng Pfizer o kaya ng Moderna, ito yung mRNA, no? So makikita yan ng ribosome kasi imagine niyo lang papasok siya sa mga cells. Papasok yan sa mga cells eh. Kasi ano siya, RNA ano, nakapasok yan sa mga cells. So pag nakapasok na siya sa mga cells, makikita yan ng ribosome. So ribosome, ano 'yon? Parang nucleo organelle siya sa cells. So makikita yan. So magbabind agad yan yung small subunit. Once na nagbind yung small subunit, no, makikita yan ng if you have mRNA, this is a tRNA, ito, tRNA, RNA. So yung tRNA, siya naman yung may daladalang mga amino acid. Of course, we're going to produce protein. Yung protein na halimbawa, ipoproduce natin, if we're speaking of the vaccine, of the mRNA vaccine, we're going to produce antibodies. Ganun, ano? So, yun. So, ito si tRNA, may dala-dala siya. Ang unang-una palagi, ang unang-unang code is AUG. I don't know kung sinadya nila to. Kasi di ba August means first. Ano, di ba Augustus is the first Roman Empire. Ganun yan. Kaya August. Yung mga months natin, mga emperor yan na naman. Si Julius Caesar, July. Ganun yan. So, it is August is the first. Kaya dun magbabind yan. Di ba? A goes with U, U goes with A, say RNA ito, Gabi conception pa rin. GC. So, yun. Ito yung first, ang first, palagi ang first amino acid ay methionine. Kaya F-met yung pangalan. Kakabit siya dito. Once na kumabit na tong tRNA na to, yan na yung signal, lalapit na yung large subunit ng ribosome, no? Ito yung large subunit ng ribosome. Tapos you will form this. Magkakaroon kayo ng E site, ibig sabihin exit site. Tapos ito yung P site, no? So dito mapo-form yung protein or yung peptide, no? Tapos ito naman yung A site, dito kakabit yung amino acid. Ibig sabihin yung tRNA na may daladalang amino acid. That's why you have the A site. No, amino acid. Ito nandito yung protein sa P site. Tapos ito yung exit site. Dito naman, dito lumalabas yung yung tRNA na wala nang daladalang amino acid. You'll see it later. Okay? So mRNA, itong red, this is your mRNA. Yung tinurok sa inyo, no? Ito yung ribosome. Nasa organelle siya. And then you have the tRNA. Sila yung naghahakot ng mga amino acid para mabuo yung protein. Yan. Science in action, Science saves the humanity. AUG. Tapos yan may dumating na another tRNA. Yung dala-dala niya ay valine, So yon. Tapos merong enzyme diyan. I don't know, it's 50 days ata, So the 50 days will form a peptide band. Alam niyo naman yung peptide band, di ba? Yung peptide band binds the amino acid. Naabot niyo ba sa sa organic chem? So it will be, tapos hihiwalay siya dito sa tRNA niya kasi naka-form siya na siya ng band. It's a much stronger band. So nandito na siya. Okay? Tapos magmo-move sila, ito pupunta doon sa E site, ito pupunta doon sa P site, tapos meron darating na bagong tRNA. So yun, may dala-dala siyang phenylalanine, So ganoon na, hanggang pahaba to ng pahaba. This is the exit site. So as you can see, lumabas yung tRNA, Yun. So this is continuously repeated until you have the UGA. This is the termination or stop codon, Kung nagsimula kayo sa AUG, magtatapos tayo sa UGA. Pero syempre, mahaba na yung series natin ng amino acid. So pag hiwalay dito ng amino acid, magiging protein na yan, So, kung sa pinag-uusapan natin ay vaccine, ito na yung magiging antibodies. So kaya effective yung mRNA kasi as you can see, yung isang mRNA, it will be able to produce lots of antibodies. Unlike yung traditional kasi na vaccine, so yung mismong patay na virus yung ini-inject sa iyo, limited lang yung napoproduce nun na antibodies kasi umaasa lang siya maasa lang siya sa katawan na magpo-produce yung katawan niya ng mRNA, tapos tsaka pa mag-undergo ng di ba? Ng protein synthesis. Ganun. Kumbaga, hindi siya diretso. May dadaanan pa siyang isa pang mag-RNA synthesis pa siya bago siya dumating doon sa mRNA. Yung Pfizer at saka yung Moderna, diretso siya dito. Yun. So basically after pag dumating na kayo doon sa UGA, ganun din kamukha sa transcription, maghihiwa-hiwalay na rin yung ribosome, hiwalay na, yung mRNA hiwalay, no? Pati yung last tRNA, pati yung release factor na ACU. This one, simulan na naman yan kung kailangan talaga ng antibodies. Magbubuo na naman siya ng antibodies. Of course, yung kasing mRNA, parang rin yan, parang vitamins din. Pag tumatagal, nasisira din siya. No? So, pero sabi naman nila, nasa memory na rin naman daw to ng system natin. Eh. Kaya lang, ang findings nila, after 6 months, bumababa na yung antibodies. No? Kaya nagbo-booster. Pero theoretically, dapat na-memorize kasi yung system natin. So, ganun yung istorya. Do you have any question regarding mRNA? Naintindihan ba? Kung paano elongate? Did you understand the elongation? So basically, nagpo-form lang ng peptide band. Probably next meeting discuss natin yung proteins para maintindihan yung mga peptide band. Peptide band is simply the connection between the chemical band that is formed between amino acids. No? So basically, lang yun. Kasi di ba COOH at saka NH3, inaatake lamang ng nitrogen yung carbon. Ganun lang naman yun. Nucleophilic. Kita nyo naman next meeting siguro. Yan, this is the elongation, So, yun, po-form siya ng protein. Do you have any question regarding, protein synthesis? We call this translation. Pag RNA, transcription, Pag DNA, replication. So, this is the summary of protein synthesis. Number 1, small subunit of ribosome bind with mRNA. Number 2, a tRNA bind with AUG codon. Ito yung may dala-dalang FMET. So number 3, big subunit of ribosome bind with mRNA and P site, A site, and E site is created. And then number 4, right here, another tRNA carrying another amino acid bind with the A site. Ito yung amino acid site, kaya A, So yung mga meaning is P is the peptide site, A is the amino acid site, E is the exit site. Ganun yun. And then number 5, peptide band is created and tRNA amino acid band is cut. Okay? 50 days ata. I will check kung 50 days yung enzyme. Pero parang tanda ko, 50 days. And then tRNA move to E site, exit site, and then peptide carrying tRNA to P site, and then a new tRNA bind with A site. Paulit-ulit lang to, itong number 6, hanggang sa humaba yung protein. Hanggang maubos niya yung buong length ng mRNA. Na-imagine nyo ba? Parang yung kasi yung yung casting niya, yung mRNA. So lalakarin niya yon, yung buo na yon. Tapos pahaba ng pahaba yung protein. Hanggang maabot niya yung UGA, yung release factor. Release factor bind with A.